today, I will be describing the high throughput crystallographic fragment screening we have carried out at Diamond Light Source on RNA-dependent RNA polymerase domain as a first step towards developing a Pan-Flavivirus inhibitor. I will briefly go through the rationale, the method that we used, the results that we got, and some of the ongoing work in this project. We have carried out this work as part of ASAP. Briefly, ASAP is one of the 9 NIH-funded AViDD centers for developing antivirals for pathogens of pandemic concern. The main goal is to deliver oral antivirals with global, equitable, and affordable access. There are many discovery projects at various stages of drug discovery pipeline within ASAP. So there are projects on coronavirus, enterovirus, and flavivirus. Pan-Flavi inhibitor of RDRP is one of the projects which is in the early stage of drug discovery within ASAP. All the information for the various projects, outputs, everything going on within the ASAP can be found in this website, asapdiscovery.org. Let me start by briefly explaining what orthoflavivirus is and why are we interested in developing antivirals against this virus. Orthoflavivirus consists of enveloped, positive sense, single-stranded RNA viruses belonging to the family Flaviviridae. It has mainly 4 members, and one of the members is a flavivirus or orthoflavivirus. Some of the members in the orthoflavivirus involves dengue virus, Zika virus, West Nile virus, yellow fever virus, Japanese encephalitis virus, and many others. An important highlight here is most of the members of this family are human pathogens. They are transmitted either through mosquitoes or through ticks. And with the current trend of globalization and climate change, there is a lot of spread of this virus with the possibility of changing from an epidemic to a pandemic status at any time. So far, there is no specific drug for the treatment of any of these infections. And so therefore, there is an urgent need for developing a Pan-Flavi inhibitor. This slide briefly summarizes the ASAP strategy. This involves focusing deep on some of these strains, like through high throughput experiments like crystallographic fragment screening and deep mutational scanning. The information from both these high throughput experiments combined give us an idea about mutationally invariant sites with initial fragment hits that can be readily used for algorithmic hit to lead development. Then we could use common interactions across the Flavi strains identified through sequence, as well as structural analysis towards developing a potent broad spectrum inhibitor. So now coming to our target, which is polymerase. Polymerase is a famous target in the sense that it has an attractive target for therapeutics for quite a long time. Polymerases are used as targets not only for antiviral therapies, but also for some human diseases like some cancers. This table briefly summarizes decades of history of using polymerase as a target. Broadly, the inhibitors developed for polymerases are of 2 categories. One is nucleoside inhibitor, another is non-nucleoside inhibitor. Nucleoside inhibitor achieves inhibition through more or less through a chain termination. These are substrate analogs. Best example is sofosbuvir, which has been developed for HCV. But the main challenges in working with such an inhibitor is off-target effects and unexpected toxicity, because nucleoside inhibitors are always administered as prodrug in the clinical trials. So the inability to predict these side effects early during in vitro testing resulted in many failures at clinical trials. The second class of inhibitors, the non-nucleoside inhibitors, work by trapping or locking the RDRP conformation. An example of this category inhibitor which has been successfully developed is tesabuvir. This has been developed for HCV infection. One thing challenging for sites away from the active site is those sites may not be highly conserved and it may be susceptible to mutation. Here, we are interested in RNA in a polymerase, which is the RNA-dependent RNA polymerase from flavivirus. RDRP is actually a C-terminal domain of the largest protein NS5 in orthoflavivirus, and it is the largest and highly conserved protein. The structural architecture overall remains the same for all polymerases, including these RNA-dependent RNA polymerase. It has 3 domains, a finger, palm, and thumb subdomain. And Flavi-RDRP carries out the genomic RNA replication through primer-independent method, which is also called de novo replication. Now let us see what are the possibilities we have to inhibit RDRP. These are the main 3 stages in the action of the RDRP. The first stage, the pre-initiation stage, the binding of the promoter to the thumb side 2 happens. So by blocking this thumb side 2, we can inhibit the RDRP at this stage. The second stage is the initiation. This is a state from which first nucleotide to up to around 6 nucleotides are added to the nascent RNA chain. At this stage, we can inhibit RDRP at 3 sites. One is the priming loop site, the other is the RNA tunnel site, and third is the cavity B site through which the RDRP interacts with another protein, NS3 helicase. The third step is elongation. This is a stage at which RDRP undergoes large amount of structural transitions. And this is a stage where nascent RNA is extended from 7th nucleotide onwards. The main opportunity here to inhibit RDRP is one at the cavity B site where the NS3 helicase interacts, and also the thumb site 2, which undergoes changes in the transition from initiation to elongation. By blocking the thumb site, we can prevent the transition. Now I will go into each of the sites in a bit detail. The first site, the RNA tunnel site, this site is located at the finger subdomain. The main opportunity here is to block the RNA template binding. By blocking this site, we can inhibit initiation, but elongation is also inhibited, but is very poor. The second site is a priming loop site. This site is located at the hinge between the thumb and the palm domains. The main opportunity here is to lock RDRP conformation. Initiation is inhibited strongly here, but elongation again is very poorly inhibited by blocking this site. The third site is the cavity B site. This occurs at the finger-thumb interface. The main opportunity here is to block the NS5-NS3 interaction. We also know from literature that mutation here impairs the RDRP activity. So by blocking the cavity B site, we can inhibit initiation as well as elongation. The last site is the thumb site 2. This is located near the C terminus of the molecule. And blocking the site blocks the promoter binding and also the structural transitions from initiation to elongation. So we can inhibit initiation as well as elongation by blocking this site. So now that we have a clear idea about the sites that we are looking for, to identify and explore these sites and to develop inhibitor, we are using a crystallographic fragment screening approach here. So what is crystallographic fragment screening? It's a highly sensitive and powerful technique for hit identification. For this fragment screening, we are using small chemical molecules with molecular weight less than 300 Dalton. And each of such libraries contain 500 to 1,000 compounds which can be readily screened. The main advantage of this technique over other technique is that this process yields the direct structural information that can drive a faster hit to lead optimization. This is the overall workflow of the crystallographic fragment screening that we followed at XChem facility Diamond Light Source. So the process starts with imaging the crystals, which is followed by compound dispensing, sample harvesting, and unattended data collection at the IO41 beamline at Diamond Light Source, automated data processing, hit identification using Panda, and model dissemination and fragalysis, which can all be completed within 2 to 3 weeks' time. The first and the foremost step in this process is preparing the sample or preparing the crystals for this high throughput screening. So sample preparation is an important step to carry out the high throughput crystallographic fragment screening. Now, When we started working on Zika virus RDRP, it turned out that the sample preparation phase was quite challenging. RDRP was a very hard protein to work with. We had to do many rounds of cloning with different levels of truncations, fine-tuned controlled expression trials, and also an optimized protocol, a protocol that has been more or less optimized at every stage of its purification to get good protein, which gave robust crystals which diffracted well and could be readily used for high throughput fragment screening. This is an overview of the fragment screening that we have done at the XChem. We have done 2 fragment screens, one on Zika virus. This was done with ASAP. The second one is dengue virus 2 RDRP. This was done with READY, which is one of the 9 AViD center. Dengue virus 2 fragment screen was done by Yasmin from the XChem team along with the members of the READY AViD center. So we got around 90 fragment hits in total for Zika, and we got around 60 fragments in total for dengue. So in the interesting site, which I mentioned in the earlier slide, we have around 62 fragments spanning all interesting sites in Zika and 44 fragments for dengue virus 2. All the fragment screening data is available on Fragalysis for Zika, dengue, and as a component overlay. Next, we checked the deep mutational scanning data, which is available for Zika NS5 RDRP. So we are looking for sites away from the active site, and so there is a tendency for this sites to be mutational variable. So to check that, a high throughput DMS scanning was performed. That is, a deep mutational scanning was performed. It's a high throughput functional assay that measures the effect of every genetic change at every site in the protein. And this work was done. The data was generated in Matthew Evans' laboratory, analyzed in Jesse Bloom's lab, and also mapped to fragment screen by Yenge Shin. So the outcome of the data is shown here. So the region shown in red are those which has lots of mutations, and the region shown in gray represents regions where the mutation is very low. Frequency of mutation is very low. So this data showed that the sites which we are interested in, there is very low frequency of mutation. Now I will go into each of these slides in detail. The first site is the RNA tunnel site. We got around 6 fragments for dengue and 38 fragments for Zika. First, we tried superposing the structure with that of the elongation complex with double-stranded RNA, and we found that the RNA passes through the same tunnel where the fragments cluster. As you can see, the Zika and dengue fragments cluster together at this site. Next, we did check the sequence of the residues forming this pocket, and we found that there are no difference in residues forming this pocket between Zika and dengue. When we compared across the orthoflavivirus strains, there are 28 out of the 34 residues conserved. There are a few residues variable between different dengue virus strains, which are shown in a red patch, and the side chain of these residues are pointing away from the pocket. The next site is the priming loop site. Here we got around 10 fragments from the dengue screen and six from the Zika virus screen. Priming loop, the inhibition mainly happens through the locking of the priming loop, and also the location of the site is also relevant for inhibition. So this site, priming loop site, overlap with the primer grip site, which binds the first 2 nucleotides in the catalysis. And also the catalytic cysteines are in very proximity to this site. Unlike the RNA tunnel site, in priming loop site, the fragments are spread over a slightly large region. Next, we checked the sequence forming the site across selected Flavivirus strains, and we found that 18 out of the 21 residues are identical between dengue and Zika. The residues which are different are shown in yellow patch here. But across the orthoflavivirus strains, 15 out of the 21 residues are conserved. One thing to highlight here is Novartis have developed a potent inhibitor acting through the site for dengue, but 2 resistant mutations developed later which reduce the potency of their best compound. The mutations that they got are marked in red patches here. And so now our task is to find a new sweet spot within this site to develop a Pan-Flavi inhibitor acting through this site. The next site is a thumb site 2. We got around 29 hits for dengue and 3 hits for Zika. In the thumb site, the dengue fragments and the Zika fragments cluster at slightly two different subsites. One thing to note here is that in dengue, this site, the dengue virus RDRP, this site comes at the crystallographic packing interface, and some of these binding would have been caused because of the crystallographic packing. Again, we compared the residues forming this pocket across the selected orthoflavivirus strains, and we found that unlike the other two sites I mentioned, this site is not very conserved. Only 15 out of the 26 residues are identical between these two sites. The residues that differ between dengue and Zika are shown in red patches here. Also, there are a few residues considered critical for the site for binding the promoter RNA. These are highlighted here in orange patch in the surface representation. And these residues are conserved across all the orthoflavivirus strains, and hence it is still possible to develop a Pan-Flavi inhibitor through this site. The last site is a cavity B site. This is the site that has been identified through mutation and functional studies, and there are no structural reports for inhibitors acting through this site. There are 14 fragments bound just to the adjacent cavity from the Zika screen. But in the case of dengue screen, a buffer component occupied this position and there are no fragments. Next, we looked at the residues forming this pocket, and we found that 7 out of the 9 residues are identical between Zika and dengue. The residues which are different are shown in red patch. But considering all the orthoflavivirus strains, we found that only 3 are conserved in all. So in summary, around 106 fragments were obtained in 4 interesting sites with good ligand coverage. And this table briefly summarizes the advantages and disadvantages associated with each site. Of all the sites, RNA tunnel seems to be the highly conserved. Priming loop is conserved, but there has been reports of resistant mutations, which we need to consider while designing a drug here. Thumb site, we have information on all inhibitions from HCV studies. We don't have much information on flavivirus inhibition studies acting through this site. Again, we need a new crystal system as this site is at the crystallographic interface in our crystals. And the last site is a cavity B. This is a site where 2 processes are inhibited by blocking one site, but there is no structural evidence for anything acting through this site. So having identified the mutationally invariant sites with fragments, our next step was to how to quickly progress these fragments. For this, we are now following a fast-forward fragment pipeline available at the Diamond Light Source. And this work is carried out by Warren, Max, Matthew, and Lisa at Diamond. So the basic idea is to augment fragment screen with one high-value, low-cost DMTA iteration. And with a one DMTA cycle, we will get chemical matter with on-scale potency, well-mapped binding site, and in a very short span of time, and also with a low-cost approach. Along with the fast-forward fragment, we also have developed a double-stranded RNA detection assay for measuring the inhibition, RDRP inhibition. This picture represents the schematic of the assay. And this basically, the fluorescence from the dye on intercalation with a double-stranded RNA that is formed as a catalysis of RDRP is measured in this assay. Currently, Charlene at Oxford has optimized this assay to perform it in a high throughput fashion using very minimum quantity of protein. And this assay can be now performed in 384 well plates with signal detected within 30 minutes. Currently, work is ongoing to standardize this assay for Zika RDRP. Now, moving on to some of the progress that we made with this fast-forward fragment work. This slide briefly summarizes the predicted poses of the compounds that are obtained from the first round of fragment merging as part of the fast-forward fragment pipeline. And these are the catalog compounds that we have purchased currently. Around 27 of these catalog compounds we have tested, and we got around 4 fragment hits, 4 hits for binding to Zika, and around 2 hits binding to dengue. The results are quite promising and exciting, and further work is ongoing to test rest of the compounds of the fast-forward progression pipeline. Now, going to the overall summary of my presentation. Through crystallographic fragment screen, we identified 4 RDRP sites with good ligand coverage, and this provides opportunities for fragment merging and growing. Fragment mergers and elaboration using AA and machine learning approaches are ongoing both at Diamond Light Source and Postera. The follow-up compounds will be analyzed through X-ray crystallography, and also inhibition will be validated through biochemical assay. And the compounds will also be tested through a biophysical assay. Now going to the acknowledgements. The work I presented here now would not have been possible without the collective effort of many members involved in various teams. And like the varied and unique experience and the expertise of the people and the knowledge has helped work throughout this RDRP project, which was quite challenging at every stage of the work. And thanks to everyone who made this project happen.